Okay, greetings everyone. Welcome to this very first session of the very first week of this algorithmic thinking in bioinformatics course. Thanks for your interest in this elective course. I know you have a lot of different options to choose from, and this says something about certain reasons that you may have chosen this particular course. And allow me to first share my thoughts also on why this is a compelling course, this is an exciting course for you to take, both for you as well as for me, right? And that is what this first session is about. It is about motivating this particular course. Why? Why do we want to study bioinformatics or algorithmic approaches to bioinformatics and computational biology? So, the rationale for the course and then rationale for bioinformatics. This is how I am going to organize this. So, as part of this program, as part of this programming and data sciences program, you have learned many algorithms and machine learning models, and you have mastered many of them in terms of the abstract representations and the algorithmic sequences, and you have worked with probably a lot of assignments on toy problems. But how do you really choose which algorithm or model to use in a real world application? So, that is really a central question if you want to translate the skills you have learned in this program so far all the way to a real world application, right. So, this is quite challenging, as most real world questions do not come with a clean problem formulation, right. So, basically you do not have a clear problem statement for a real world question, and also you do not have a obvious algorithmic solution, because you do not have a clear problem statement, you cannot even think about what are the algorithmic solutions for the problem. Fortunately for us, this challenge could be addressed by taking a particular applied area, a particular specific data science field, and then trying to see how the algorithms and machine learning models are being used in that field, are being applied and adapted and made to work in that field, right. And this is the main premise of this course is you can learn how to solve these real world questions using programming and data science analysis by basically observing these algorithms in action, right. And the particular field we have chosen for this course is bioinformatics and computational biology. And then within this particular data science field, we want to be able to understand different aspects of how you would go about solving a real world question, including problem formulation, right. Like converting a real world question into a particular clean problem statement, a clear problem statement. How do you choose which algorithmic paradigms to use? So, you have again seen many different paradigms in the course, dynamic programming, greedy algorithms and other things along the program in different courses. You have seen different machine learning techniques. So, how do you choose among these algorithmic paradigms or machine learning techniques for the particular problem at hand, right? And then what are the key data structures to represent? That would be as part of the algorithmic choices. So, these algorithms, for them to be efficiently run, you need to choose the correct representations of your data. So, how do you choose these key data structures to represent the big data sets that are coming up in this particular field that we are going to focus on in bioinformatics and computational biology. And so, then I will have to motivate you on why we chose this particular field, right. There are many other fields, from finance to social network analysis to other areas. So, why this particular field of bioinformatics? So, that is what we are going to look at next. So, and it is actually quite easy for me to motivate, because bioinformatics or computational biology has really addressed many valuable questions in biology and medicine using algorithmic approaches. And this power of computational or algorithmic thinking that is really an understatement, because to really say what it has done is it has really transformed the field of biomedicine in recent times, because biology and medicine has become more and more of an information science these days, and computation has played a very key role in transforming the field as it stands now. And what do we mean by this transformation, right? So, what do we mean by this computer science or computation has transformed this field? These computer science has been used, many algorithms or models like machine learning models or graph algorithms or tree algorithms or string algorithms have been used to understand different biomedical data sets. And these data sets are typically, we call them as big data in biology. These are coming from very large scale high throughput experiments. So, there are lots and lots of data that are being generated that it will never be possible for an experimental biologist to just look at the data and then figure out what the insights are from the data, to extract insights from the data without the help of a computational analysis of the data, right. And this is only becoming more and more needed, more and more inevitable, because like I said, there has been huge advancements in the technology for generating data from biology, and they are leading to larger and larger data sets, which means we need to come up with more and more efficient algorithms and models to analyze and extract biological insights from this data. And because this has been around since the 90s, right. So, the early 90s or early actually 1950s is when the first protein sequence was figured out, the sequence of amino acids that make up a particular protein molecule. In this particular case, it was the insulin molecule that was figured out. Since then, there has been rapid advances in the kind of algorithms and data structures used in bioinformatics. So, there are many examples for us to look at and learn how data science is practically applied in this particular field. And that is in brief on what I wanted to say about why bioinformatics, but let us delve into it a little bit more in detail. Okay. That is what my focus on the next several or next tens of minutes would be, right. So, let us more carefully dissect the reasons why you may be excited about or motivated about this particular field, and especially this interdisciplinary field between biology and computer science, right. So, why computational biology? I am going to use the term computational biology or bioinformatics interchangeably in this for the purposes of this course. So, let us just answer the question, why do we want to study computational biology? The first reason or the first set of reasons is practical. People usually recommend, okay, what do you want to do in your life? You say follow your heart, but then some are prefixing that with pay your bills, get your finances right, then follow your heart, right. So, life sciences, so let us look at certain practical reasons why you might be interested in learning about bioinformatics or specifically life sciences, right. So, applications of computational or algorithmic thinking in life sciences, which includes basic biology as well as healthcare, medical data analysis and so on. So, life sciences is a fertile ground to practice and gain end to end data science skills. So, like I said, it encompasses different fields. So, there are many companies in this space, starting from pharmaceutical company which are involved in drug or medicine discoveries, to structure prediction of different molecules, to medicine that is not given the same tablet given to everyone, but you want to personalize or target it for certain groups of people, you want to give different medicines for the same disease, right. And how do you understand the different molecules, genes and other things that are involved in a particular disease. In our human body, we have 20,000 genes, and not all genes are involved in all the diseases. So, different genes have different diseases have different genes which are being affected. So, how do you get to find those target genes for a particular disease? So, all of these there are certain bioinformatics solutions for them, and so pharmaceutical companies have put bioinformatics to good use to solve these problems. And there are diagnostic companies. So, not just those that are discovering new medicines or targeting medicines for certain groups or certain individuals, as in precision or personalized medicine. You may also want to look at diagnosing if a particular person has a risk for a certain disease. So, those are genetic panels that you can use to measure different individuals and build machine learning models to predict what is the risk of a certain disease, right. And there are early discovery or early diagnosis of diseases such as cancer and subtyping of these diseases. So, many diseases do not come in a single flavor, there are different subgroups of diseases, and so you have disease types and subtypes. And so, how do you identify different subtypes of a disease? And that is a classic clustering problem, if you will. You are given this patterns of diseases and you want to identify these different subtypes or sub patterns that divide or cluster a disease into multiple different sub diseases. One example is autism is actually a spectrum of different disorders. So, how do you figure out which spectrum you are in based on certain measurements or data that you collect from the individual? And then there is literature mining. So, biomedical literature, there are about 23 million articles, biomedical research articles in PubMed alone, and then add to it like clinical trial documents, patents and other things, and then you have a huge corpus of biomedical literature. And can you apply natural language processing, NLP, and other related machine learning models to extract insights from that literature, research literature, electronic health records, patents, or clinical trial corpus, right? And this is all the reasons in terms of practical reasons in terms of which are the industries or companies that are involved in life sciences, and there is transferable skills. So, even if you are not interested professionally in this particular life sciences domain, the skills that you draw from this particular studying this particular field of bioinformatics or computational biology could be transferable to other domains of interest. Okay. And basically you want to draw, I mean, what kinds of skills you may ask. So, these are concepts or ideas we will draw from almost every earlier course you may have taken in this program, like every foundational course such as algorithms to basic statistics to machine learning. Okay. And the other category, so those are the practical reasons why you may be interested in bioinformatics and computational biology. The other one is more in terms of impact, and let me ask a question. So, what is the first algorithm or machine learning model to touch a human's life, right? So, in the or to put it another way, right, in the lifespan, right, of a human being. So, what would be the first ML model that is used? Something that happens that even before we are born, we are touched by a machine learning algorithm, and that is ultrasound based fetal weight estimation, right. So, you can have these are some of the linear regression models. So, you have again seen linear regression earlier in this program. So, this is a linear regression model which looks at different parameters from the ultrasound images of a fetus, right. So, baby before it is born is called a fetus. So, you look at the fetus growing inside a mother's womb, and then you look at ultrasound measurements, and you look at the diameter of the head, for instance, that is called BPD over here. Let me just point it out. So, the BPD here is the diameter of the head, right, and then AC is the circumference of the abdomen. So, using those two measured in centimeters, right, this is a model. Then if you just replace with AC squared, BPD squared, all of that with those measurements of the baby, right. This is even before the baby is born, right. So, these are the fetal BPD and fetal AC, the fetal head diameter and the fetal abdominal circumference. Then this formula will give you the weight, the estimated weight of a baby, the predicted weight of a baby in grams, right. So, this is a model, a machine learning model that touches our human lives even before we are born. And another example is also along similar lines. So, the ultrasound imaging is non-invasive, you just do not touch the womb or go inside the womb to extract any samples, right. So, you can diagnose rare disorders like trisomy, which are basically extra chromosomal copies. So, our DNA content within our cells are organized in the form of chromosomes, and we have certain number of chromosomes that we have inherited from our mother and father. But in trisomy, which is a disorder where we get one extra copies of certain chromosomes. So, there are 23 pairs of chromosomes in our human body, and then so if you have chromosome 21 or 18 or 13, where instead of 2 copies, one from mother and one from father, you have 3 copies, then you have issues, you have disorders, and these are called Down syndrome. If chromosome 21 has an extra copy, Edwards syndrome if you have 18, and Patau syndrome. You can draw a blood sample from the mother's blood, right. So, you are not going and touching the womb or anything, you are just extracting blood from the mother, taking a blood sample from the mother during pregnancy, right. And then after certain weeks of pregnancy, so the blood in the mother actually contains genetic material. So, the DNA of both the mother of course, because there are many cells in the mother which may be shedding DNA into the mother's bloodstream, but also from the child, right. So, those are shown as the blue colors here. So, there is also a fetal DNA that is floating around in the blood, and by looking at that DNA, then you can figure out. So, you can then do these experimentation. Like I told you, there are these huge advances in the amount of data you can gather from these samples. So, there is some technique called massively parallel sequencing, where you can convert this blood, you can go through certain processing and then actually read out the letters that make up these DNA reads, right. So, this DNA. and then you can figure out and then do certain. So, this arrow is where bioinformatics analysis happens. So, this arrow is where experimental processing happen, right. So, you have all these experiments which are converting this blood sample into these sequences of strings. So, there is experimental way of reading of these sequences, and in these instruments also there is bioinformatics analysis happening. But once you have these sequences, you also have to then do certain statistics and counting to figure out how many copies of these different chromosomes are there, right. So, blue is chromosome 21. In a normal person and a normal fetus you will have 2 copies, but in the in this kind of chromosomal extra copies you will have 3 copies, right. Trisomies you will have 3 copies. So, next generation sequencing, which is one form of massively parallel sequencing, is an experimental technique for reading of these letters of these ACGT which make up our DNA or which make up the fetus's DNA, and bioinformatics analysis of that is used to identify these chromosomal disorders, which are which like Down syndrome are serious that you would like to know about it before than later on, right. So, this is the reason I say that this is impactful, that even before we are born there are bioinformatics analysis or computational biology actually impacts our lives, human lives. And then let us go on to something that is very closer to my heart, in the sense of what are the scientific or research reasons why you may be interested in bioinformatics or computational biology, right. We have seen practical reasons, we have seen the impact it has on human lives, and I know that as all the students who are taking this course, you may have different interests ranging from life sciences to finance to other forms of industries. So, even if you are not into research area, I just want you to keep an open mind and just be silent and listen, right. Like William Arthur Watts says, before we learn we will have to listen, and before we listen we will have to be silent, right. So, let us just silence our doubts on whether I really want to go into bio or not, and then let us just listen what I will have to say. Okay. So, in terms of the scientific or research reasons, let us see why we do interdisciplinary coursework or research like computational biology in the first place within computer science, right. So, that is what the next 5 or 10 minutes I am going to look at. Okay. And the basic answer is this: many real world questions, think about climate change for instance, cannot be solved by one discipline. So, these disciplines or fields are all thought about by humans, they are artificial constructs, so that we can easily master a particular area and it is tractable for us to learn different areas. But real world questions do not care about what humans we have come up in terms of this is physics, this is chemistry, this is biology and so on. So, the real world questions just pop up, and then depending on the question, many complex questions involve looking at different areas. And the early researchers were naturally cross disciplinary. They I mean at that time there was not very specialized disciplines. So, it made it easier for them to be masters in multiple fields, and they were able to address these questions. So, Alan Turing, you would know as the father of one of the founders of computer science, right. He actually worked in both computer science and biology. So, you know his major contribution is Turing machines amongst other things, and also the Turing test, right. He touched both in terms of foundational algorithms as well as machine learning. I mean, so Turing machine is one such I mean main contribution of him, and then he also studied chemical basis of morphogenesis, which is embryo development. Ronald Fisher, again in your foundations of statistics you may have heard about the maximum likelihood. So, he contributed in a major way to maximum likelihood theory, and at the same time he also looked at evolutionary problems in biology. Max Delbruck is another example who I mean another researcher who worked in both physics and biology, right. And this is a quote that I really like. I mean many of my students would already know by now the answer to this question, who said this quote. So, there is a person a very eminent researcher who said basically that biology easily has 500 years of problems, open problems, exciting problems to work on. It is at that level, but he or she, this person was not confident about his or her own field, right, whether there is unending growth in that particular field, right. And I am going to ask if any of you know who this quote is by. Okay, maybe let me ask a question, would it be by a computer scientist or a bioinformatician or a biologist? Computer scientist, yes. So, so this computer basically this person is not sure about the kind of the unending growth in computer science, but then shares that biology easily has 500 years of exciting problems to work on, and this is actually by Donald Knuth. And you know him as a classic computer scientist. I mean, who can get more classic than Donald Knuth, right? I mean, his books are so popular and famous. and he has made seminal contributions. I mean, for those contributions he was awarded the Turing Award, which is like the Nobel Prize equivalent in computer science, right. But he said this in 1993 that there is easily 500 years of exciting problems to work on in basically biology. At that time though, maybe he did or maybe he did not, he may not have been able to see the tremendous growth that bioinformatics or computational biology would have achieved. So, it started out as an interdisciplinary area. Now, you can even say it is almost a discipline on its own, and it has merged concepts from computer science and biology to take this forward, to take this kind of addressing problems in biology using, or whatever problems are amenable to solve using computational analysis. It has taken major advances, right. And it has even in 1990s the computations were used, because remember that the first insulin protein sequence was sequenced in 1950s. So, even in 1990s some amount of computational analysis of sequences was already quite popular, and then there was a human genome project around the 1990s. So, which could have also spurred this kind of comment from Donald Knuth during his interview. But then why is this renewed focus, right. So, we have continued, I mean since then we have continued this renewed focus on interdisciplinary research, and then that really the basic premise I am putting forward is modern biology is inherently quantitative. It has become more and more data driven, right. So, it has become more and more of an information science, and because of that it is often crossing disciplines, right. What do I mean by that? Here are some illustrative studies. I am not going to give you a formal reason, but some illustrative studies on why modern biology has become more and more quantitative, right. So, basic research into molecular mysteries of life, right. So, those are some set of studies that bioinformatics or computational biology has solved, right. Here again I am thinking about it in a broad sense. So, there is biophysics, there is biochemistry, and so there are kind of different fields that are all trying to answer questions in biology. Anything that involves analysis of data, modeling of the system, using a computer to solve it, I am just grouping it under biology, I mean bioinformatics and computational biology, right. So, there is a paper in 2012 by Carr et al. Basically they took a whole cell and then came up with a mathematical model of all the major biomolecules within the cell and how they interact with each other, how they control each other, how they regulate each other. Right. This was a major breakthrough for a particular cell, and then so there is a lot of measurements involved, lots of data involved, analysis of the data, modeling of the data using a multitude of different models, from ordinary differential equations to certain other statistical analysis, right. As one example. The other example is if you do not want to look at a whole cell, but you want to look at particular process within a cell, the process of how genes get activated. Like I said, we have 20,000 genes within our human body, but not all of them are active all the time in every cell, right. Different subsets of genes are active in different cells, and so how do those subsets gets determined, and what are the levels of the genes that are active, and so that was modeled in great detail using actual imaging and other measurements in this particular paper by Batish et al. Those are from basic science, right, basic science or research, and there are medical applications, so illustrative studies from medicine, where this is one of the early this Golub et al, right. This is one of the early successes in computational biology or bioinformatics, where traditionally if you want to understand different types of cancer, if you want to subtype a cancer, like if you want to look at what exact type of cancer it is, you look at a biopsy or a sample of a cancer, look it under a microscope, and then look at what are the different morphology or the structure of the cancer tissue, right. But then here they were able to look at the levels of genes, of different genes in each of these tissue, again using this technology for measuring all the genes in the genome, the activity of all the genes in the genome in different tissue samples, and use that measurement. So, here the rows are different genes, and then the columns are different individuals from which the samples are obtained, right. And then blue means that particular gene is low level in that particular individual. So, let us look at this last gene, which is catalase. Catalase is red in this last set of individuals, right. It's highly active or expressed, as another term we use is expressed. So, this gene is present in high levels in the last set of individuals, but low levels in the first set of individuals, right. Similarly, all these genes are present in high levels in the first set of individuals, which are this ALL, and then low levels in the second set of individuals, which is AML. Okay. So, ALL and AML is a label that has been added later on, this is just different types of a particular cancer called leukemia. So, there are different, and they may look similar from under a microscope, from a very crude level, right. But then by getting into a more molecular level, it's quite easy. You can see that there is a quite stark distinction between these two subtypes of cancer, right. These two types of leukemic cancer. Leukemia is basically a blood and bone marrow cancer, and so this is affecting one type of cells in the blood, and this is affecting another type of cells in the blood, right. So, I mean, this has been followed by many studies where cancer subtyping is done using molecular level information rather than just using microscopy or kind of imaging information alone, right. So, that is looking into the molecular data generated from these samples. How can you understand disease sub diseases, right, classes of diseases differently, better, subtypes of diseases, and this has been followed up by this TCGA, which generated not only this gene expression data, this activity level of genes as shown in the left side, but also different other data like microRNA, mutation. These are genetic data. MicroRNA is another form of genes, shorter genes, right. So, all of this data are combined together along with clinical data to figure out how to develop better diagnosis of different subtypes of cancer, and to develop targets or discover targets that can be used for better development of drugs and therapies. And then in recent research, I will just say, keep this brief. Genes are just one part of biomolecules within a body. So, there are different small molecules called metabolites, which are also important for the functioning of a living organism. So, there are models that try to basically extract all forms of information from the literature on how the different metabolites are involved in metabolic reactions and how energy is being processed and used by the body. And then there is a whole body metabolic model, for instance, involving 30 plus organs, right. So, that is this work by Thiel et al. It is called Harvey and Harvetta. Harvey is a model for male metabolic reactions, and then Harvetta is a model for female metabolic reactions. And then this one I do not have to say. I mean, COVID pandemic has really affected our lives. I mean, thankfully not too much more these days, but then in the initial waves, it has had a major impact on human population across the globe, and sequencing and a lot of data analysis and epidemiologic analysis was done to understand where it originated, how it spread, what are the strains, what are the resistance of these strains. I mean, are they responding to vaccines and so on, right. Okay. So, to summarize this part of scientific or research aspects of why you may be interested in bioinformatics or computational biology is this particular interdisciplinary area, as this sits right in between computer science and biology, right. So, it is in the spectrum between computer science and statistics and biology and medicine. You have bioinformatics or computational biology, and interdisciplinary research is really how human scientific endeavor started in the lines of Turing and Fisher and others. And like I said, the premise is that modern biology has indeed become more quantitative and data driven, and modern computer science is also interested in looking at the interface to these other sciences which are more data driven, right. And because of that, many of these fruitful efforts are being done, and also many real world biomedical questions are also inherently interdisciplinary. Okay. And here is one particular example of why we want to push, right. So, this circle represents all of human knowledge, let us say, and then if you are pushing along this direction, you are trying to extend human knowledge along this direction along a particular discipline, computer science, and here you are trying to push it along a particular discipline, biology, right. You may want to be interested in also pushing along this interface between the two, because these real world problems, like I said, often involves advances in both of them. So, this particular knowledge may be just outside the reach of our current human knowledge. So, this is just a arc that is taken from here, right, this arc, and then there is one particular knowledge that we need to save a particular human life or to answer a fundamental question about nature, right. And if you want to look at an example of how sequencing and data analysis saved a particular son's life, right. So, this author's son's life, then this is a good read. Okay. How sequencing data analysis was used to figure out a particular mutation in a son, in this author's son, and then that was then being used to come up with certain ways to manage the condition of his child. Yeah. Okay. So, to summarize all these different categories that I have said so far, the first is practical. So, life sciences is a fertile ground to practice and become a data science expert. So, those are transferable skills that you can take it to any other field, or there are huge number of companies in the life sciences area as well, right. Then it is impactful. So, biomedical data science can help develop better health diagnosis and disease cures, and it touches our human lives before even we are born, right. And scientific and research, we talked about in length about what better pursuit than unlocking the molecular mysteries of life, right. And with that I wanted to add two more, which is favorable priors, right. As diverse audience, you are all coming from different areas, and then you have chosen to try out this course, right. To learn more and grow. So, there is no one, this is not a core course that you are forced to take. So, there is some kind of inherent interest you have. It may be overlapping with some of the interests that I shared, or it may be a distinct interest that you yourself bring to the table. So, that makes it easier for a teacher. The teacher could not wish for more rather than inherent interest in the area, right. So, there is a favorable prior in terms of starting interest that you bring to the table. And then also I also get naturally excited about works at the interface of computer science and biology, and so that is a favorable prior that I bring to the table, and I have experience from different all the way from my PhD to other industry and academic experiences that I bring to the table in terms of the enthusiasm about working in this area that I hope to share through this course. Okay.